Let's talk about the steps involved in the procedure for precipitation of proteins using ammonium sulfate. The first step is to decide what kind of beaker or what kind of glassware is going to be used. Kitni capacity ka glassware chahiye. The capacity will be decided keeping in view the required the volume of the sample. अगर हमारे पास 1 liter sample है protein का, we are going to use 2 liter volume beaker. Take your protein sample or homogenate in the glass beaker, and for each 1 milliliter of the sample we need half gram of ammonium sulfate. Keeping in view the volume, weigh down the ammonium sulfate, and if it is not in powder form, then using mortar and pestle grind it, क्योंकि अगर ये lumps की form में use करेंगे तो solubility का issue आएगा. It will take more time to get dissolved. Then we have to shift the beaker on ice so that the temperature can be maintained, and we start adding ammonium sulfate in steps. Don't put or pour all the ammonium sulfate at once in the beaker. In steps it is added into the protein extract, and at the same time we are going to stir the sample. अभी ये जो छोटे छोटे बैचेस में हम अमोनियम सल्फेट पोर कर रहे हैं एंड इसको डिज़ोल्व कर रहे हैं, पहले बैच के कंप्लीट डिज़ोल्व होने के बाद अमोनियम सल्फेट का दूसरा बैच ऐड करना चाहिए. इससे क्या होगा कि सॉल्युबिलिटी जो है दैट इज़ गोइंग टू बी ईजियर एंड क्विकर. अदरवाइज द सॉल्युबिलिटी may take time and we may end up having some salt at the bottom of the beaker. उसके बाद जो ख्याल रखने वाला पॉइंट है कि stirring should be slow, because if we are going to stir the sample vigorously, then keep in mind हमारे sample में ट्रिटोन X100 था जो कि फोम बनाता है. फोम बनाना आसान है, उसको बाद में सेटल करना थोड़ा सा मुश्किल हो जाता है. सो स्टिरिंग शुड बी स्लो डाउन, नॉट सो फास्ट. वंस वी कंप्लीट द एडिशन ऑफ ammonium sulfate in steps and we make it sure that all the salt has been dissolved, the next step would be let the sample for 10 minutes as it is so that the precipitation can be completed. अब इस 10 minutes time में क्या हो रहा है, at the back end ammonium sulfate is going to be dissolved by the water molecules. The hydrophobic parts of the proteins, they are exposed, they are interacting with each other and they start getting precipitated, then they are visible. Once the precipitation is complete, the supernatant or the liquid present in the beaker, it contains water and ammonium sulfate, which can be separated by centrifugation at 5000 G for 30 minutes. और इस centrifugation के लिए हम फैल्कन ट्यूब्स यूज़ कर सकते हैं जो कि स्क्रूड कैप के साथ आते हैं. उनके वॉल्यूम के सामने रखते हुए वॉल्यूम डिसाइड किया जा सकता है whether we are going to use 50 milliliter tubes or we are going to use some larger tubes. After that, the supernatant is decanted, is separated, and the precipitated proteins, they are going to be dissolved in our required volume of water or any appropriate buffer जो कि आपके इस्तेमाल में है. And in this way we can reduce the large volumes into small volumes, having all of our required proteins in these small volumes. At this time we have to think about that still some of the high salt concentrations are present in the sample which needs to be removed. For that purpose we may use these additional steps. The additional step is the dialysis step. For dialysis step we need the dialysis tube, and that dialysis tube is filled up with the protein sample. और उसको फिलअप करने से पहले एक एंड से सील कर लिया जाता है, दूसरे एंड से उसमें सैंपल लोड किया जाता है, फिर दूसरे एंड से डबल नॉट के ज़रिए सील करने के बाद उसे हम बफर में डिप कर देते हैं जो डायलिसिस ट्यूब है। अब बफर में डिप करने के बाद जो सेमी परमेबिलिटी कॉन्सेप्ट है ट्यूब का, उसकी बेस के ऊपर अमोनियम सॉल्ट एंड सल्फेट दे आर पासिंग थ्रू दी dialysis tube and the sample is dialyzed. 2 times dialysis step को follow करना चाहिए ताकि maximum salt can be removed, and this dialysis buffer will be can be decided on the basis of your future step. What is the next step? If you are going to perform the column chromatography, the column buffer है that can be used as dialysis buffer. After dialysis, we can perform the centrifugation of the precipitated material, and we can use that protein for any further study or for any further method.